I need to. Okay, we talked about the energetics, we talked about the enzymes, we talked about the enzymes a little bit in the context of cellular respiration when we talked about the first reaction of glycolysis. So let's talk about the mechanics now. And let me put the equation up here so that you put it in your minds. Okay, so we're going to show how cellular respiration takes the free energy in glucose and makes somewhere in the region of, I'm just going to say 34 ATP. Okay. It's a range, but I'm going to say 34. Okay, so I've got to account for the energy, so I'll do that. And I'll also account for the matter. The matter that I really care about to start with is the carbon. So you guys heard this before. How many carbons in glucose? 6. Okay. Okay. How many must I get out of this end somehow? 6. I must get 6 out. Yep. Okay. So I'm going to get 6 out over there. So that's what we're looking for. The 6 carbons in here ending up in the 6 carbons in carbon dioxide. They've got to end up in that because there's no carbon in water, and that's all we get out at the end of the day. So it's all about energy balance management, and it's all about matter balance management. So that's what we're going to focus on to start with. Okay, so the first set of reactions is called glycolysis. And so we'll start with 1 glucose. And everything I'm going to do is for 1 glucose. Okay, so if you can do it for 1 glucose, you can do it for 10, for 20, for a million, for a mole, 1.5 moles, whatever. Okay, so the first set of reactions called glycolysis convert glucose to a molecule called pyruvate. So don't forget, we've got 6 carbons over here. These have 3 carbons. So how many pyruvates must I get if we don't lose any carbon between the start and the end here? Let's get 2 pyruvates, because 2 times 3 is 6. Okay, so our ultimate goal, remember, is to make all that ATP. We're going to make a little bit of ATP when we start off. And we're going to start off by making 4 ATP. But to make those ATP, we actually have to put some energy in the form of ATP. So we put 2 ATP in, and we get 4 out. What's the net gain now? Net gain's 2, yeah? Because these 4 minus the 2 we put in, and that gives us a net of 6. So let's have 6 ATP down here that we can keep track of. Okay, just keep track of the important molecules right now. There's 6 ATP. Oh, that should be 2. I'm thinking ahead to carbons. 2 ATP. Okay, so we've made a little bit of ATP. That's great. It's not that great, but it's a good start, I guess. Okay, the other thing that we're going to do to start with is we're going to take some electrons from glucose. So if I'm taking the electrons from glucose, am I reducing glucose or oxidizing glucose? I'm oxidizing it. So I'm going to take electrons from glucose, and if I take electrons from glucose, I've got to give them somewhere. I'm going to give them to this molecule NAD+. And when I do that, I'm going to get this molecule NADH. Now, it's hard to visualize electrons moving. I can show you if we drew the structure of this, but it's easier to follow not electrons, but to try and follow hydrogen atoms. So that's easy to see. If you think about adding in a hydrogen to here, you get this molecule NADH. So for each of these glucose, I need 2 NAD+, and I get 2 NADH. It's really critical that if I'm getting NADH out, I must put NAD+ in. Otherwise, I'm not maintaining matter. I'm destroying or creating matter. I can't just create NADH and not put NAD+ in. So if I don't draw the NAD+ in, that's a violation of the first law of thermodynamics. So I must always have something going in and something coming out. So we've got a little bit of NADH now. This is going to become a very important molecule because it's the molecule which is going to store electrons from glucose. Now you remember when we talked about the types of bonds which contain a lot of chemical potential energy? Was it the polar covalent bonds or the non-polar covalent bonds? It's the non-polar covalent bonds. Okay, so the non-polar covalent bonds in glucose, we're going to take the electrons from there and we're going to put them in non-polar covalent bonds in NADH. Okay? And so as we do that, we're just shifting some free energy in the form of electrons from glucose to NADH in the bonds we form. So remember, when you break a bond, energy goes in. When we form this bond, So energy, you can put energy in, but when we form the bond here, we're actually transforming some energy from glucose into NADH. So this is going to become critically important, and we've made 2 of our 34 ATP, so a 17, which is pathetic. Okay, the next thing we're going to do is we're going to move the pyruvate into the mitochondria. Okay, so this was my first stage, it's glycolysis. My second stage is I'm going to convert these 2 pyruvates to 2 acetyl-CoAs. Okay, so 2 of those and 2 of those. Now these have 2 carbons. So if I've got 6 carbons here and I've got 4 in total here, how many carbon dioxides must come out? I've got 6 carbons going in and only 4 preserved in acetyl-CoA. So 2 must come out. 6 minus 4 would be 2. So for every 2 of these acetyl-CoA I move in, so 2 of these pyruvate I move in, I get these 2 carbon dioxides. Think about it as a molecule of pyruvate goes in, carbon dioxide comes off. That's 1. The second pyruvate goes in, another carbon dioxide comes off. That's 2. Okay, so I get 2 carbon dioxides out. But also, as we move a pyruvate in, we're going to oxidize it, and that's what's going to give us some of the acetyl-CoA. So we oxidize this pyruvate, we give electrons to NAD+ and make NADH. So each of these pyruvates gives us an NADH. So 2 pyruvates means 2 NAD+ go in and 2 NADH go out. So we can add in another 2 NADH. Yep, there's another 2 NADH there. Makes sense so far? That's the second stage. First stage, glycolysis. Second stage, conversion of pyruvate to acetyl-CoA. All the time we are oxidizing the starting glucose. So right now, I've got 4 NADH, I'm going to make 10. I've got 4 of the 10, okay, so that's 2/5. And I've got 2 ATP, that's a 17. So I don't have much of the energy that I had to start with harnessed in these molecules. So where's the rest of the energy? It's in the acetyl-CoA. It's got to be in here. It's not in these NADHs and these ATPs. It's got to be what we've produced so far. So it's in acetyl-CoA. So the third stage is going to continue the oxidation process. It's going to take that acetyl-CoA and through a series of 8 or so reactions in a cycle called the citric acid cycle, or the Krebs cycle, or the tricarboxylic acid cycle, we're going to start making some more of this NADH and we'll make a bit of ATP. So let's do 1 acetyl-CoA at a time. 1 acetyl-CoA goes into the citric acid cycle. 2 carbon dioxides come off. Okay? 1 acetyl-CoA goes in, 2 carbon dioxides come off. For each of the acetyl-CoAs, we can do enough oxidation to reduce 3 NAD+ to NADH. So I get 3 NADH out. So let me just draw a purple box around that so you can keep track of that. There's 3 NADH there. Okay. We also get a small amount of a molecule called FADH2. And that's made from the molecule called FADH. Remember, if I get something out, I've got to put something in. So I get one of these FADH2. FADH2 is just like NADH. It's structurally different. It's not as high energy. All it does is carry electrons up. Okay, so just like NADH, it's carrying these high energy electrons. Rather than taking them from glucose like we had here, or from pyruvate like we had here, it's taking them from one of the intermediates in here. The last thing we're going to get out is per acetyl-CoA, we're going to get an ATP out. And if I get that out, I've got to put an ADP in. Now your book says this is GTP, which is correct, but the GTP is immediately converted to ATP. So I'm just going to put ATP because I think it's simpler than worrying about the GTP. So if I get all of these for 1 acetyl-CoA, how many carbon dioxides do I get for 2? 4 carbon dioxides. How many NAD+ must I reduce? 6, yeah. Yeah, you get it, it's okay. You attended it, but you get it. That means I'm going to get 6 NADH. If I put 1 FADH in per acetyl-CoA and I've got 2, how many FADH do I have to put in? 2. How many FADH2 do I get out? How many ADP go in? And 2 ATP come out. So, by the time we've done this set of reactions, we've got another 2 ATP here. So let's count them up and write down what we got. We've got 6 NADH plus 2 plus 2. What's the total of that? 10 NADH. How many ATP so far net? 4 ATP. 2 here. And the net gain of 4 minus 2 is those 2 there. So 4 ATP. We get 1 FADH2, so we get 2 FADH2, and we get 4 carbon dioxides there, 2 carbon dioxides there, for a total of 6 carbon dioxides. I've accounted for the carbon dioxide, and I've accounted for what's happened to the glucose. All I got to do now is count for the oxygen and the water, and then I'm good. Where is the energy now? Is it in the carbon dioxide? You wouldn't be exhaling it if it was useful yet. You'd be breathing it back in again. So, this is where the energy is at this point. So these are just alternative forms of chemical potential energy. So what we're going to do is get the energy out of the NADH and FADH2 to make an additional 30 ATP. So I've got 4 already, and I said I'm going to make 34, so I'm going to make another 30. So the last set of reactions is where we make the rest of this ATP. So let's go back and go glycolysis up to here. Conversion of pyruvate to acetyl-CoA up to this point. The Krebs cycle. And the last set of reactions is called oxidative phosphorylation. It's broken into 2 stages. The first stage involves what's called an electron transport chain. And the second stage involves an enzyme called ATP synthase. So this is all going to happen on the inner mitochondrial membrane. So remember, this is occurring in the mitochondria, yeah? So all of this NADH and FADH2 is being made in the mitochondria. This makes sense because we're now going to do the rest of the reactions in the mitochondria. So this is just a little piece of the mitochondria. Kind of looks like the edge of a mitochondria, right? Outer membrane, inner membrane, we've got the matrix, and we've got the space between the 2 membranes, the intermembrane space. If that isn't in your head, you've got to take a step back and you've got to go and review mitochondrial structure. Because when I write questions for this, I will use this language of outer membrane, inner membrane, intermembrane space, and matrix. If you don't know what those things are, you can't interpret the question. So you've got to go back and make sure you understand mitochondrial structure. Okay, so the electron transport chain does exactly what it sounds like. It transports electrons. And the proteins are arranged in a, for want of a better word, in a chain. So let me draw a chain of proteins in the membrane. Okay, I'm going to draw 4 to start with. And these proteins have got fancy long names which you don't have to worry about. We're just going to call them complex 1, complex 2, complex 3, and complex 4. As well as these proteins, there are going to be some little shuttle proteins which we'll talk about as well. So I'll just draw those in as well. Okay, so the point of this is to transport electrons. Where are the electrons? We took them from glucose and we put them where? NADH and FADH2. That's what these molecules do, yeah? They're electron carriers. So we've got to get the electrons out of those. So let's start with NADH. NADH, if it gets close to electron, to complex 1, its electrons will move into complex 1. And if we take electrons from NADH, we get NAD plus back. It's just the reversal of this thing, yeah? If you do that backwards, you get your NAD plus back. So this NAD plus is used here, here, and here. So if you can't do this, you can't run a bunch of that stuff. So you've got to be able to do this. So the electrons go into complex 1. The reason they go into complex 1 is that complex 1 is more electronegative than NADH. Remember we talked about electronegativity at the beginning of the semester? That's where an atom wants an electron more than another atom. So oxygen is more electronegative than hydrogen. That means oxygen really wants the electrons. So complex 1 is more electronegative than NADH, so the electrons go in. The electrons can then be passed through the chain. But when the electrons go from NADH to complex 1, they lose a little bit of energy. Whenever you move electrons, there's a loss of energy. So when we did these electron movements, when we took the electrons from glucose over here, we lost a little bit of energy. So there are some inefficiencies. There's always some energy that's going to be released. The energy that can be released is used by complex 1 to pump the protons into the intermembrane space. So the work being done by complex 1 using the energetic electrons from NADH is to pump protons. The electrons then get passed to a little shuttle protein, and I'm just going to draw the electrons as 2E's. The electrons get passed to a little shuttle protein, and then that little shuttle protein can move through the membrane and give its electrons into complex 3. And when they move into complex 3, energy is released and protons can be moved into the intermembrane space. Okay? The electrons then get passed to another little carrier protein, and then to complex 4. And complex 4, again, when it gets those electrons, a little bit of work is done, because there's some energy released, and the work is the pumping of the protons. So if I'm pumping protons from the matrix into the intermembrane space, is the pH of the intermembrane space higher or lower in the matrix? Lower, because high proton concentration, lower pH. Okay? That's the kind of question that's going to come up. It ties together the principles of active transport, pH, concentration. So when we get to a complicated concept like this, I can use some other principles to see how well you understand this. So the electron movement here through the chain releases energy. Okay? And that energy is used to build this proton gradient. So that proton gradient can be used to do some work in itself. Remember we talked about secondary active transport, and we used ATP to pump solutes, and then we let those solutes come back down their gradient, and when they came down their gradient, like water coming down a dam, we were able to move another solute up its gradient. So this gradient contains potential energy. So what we've done is we've taken the chemical potential energy in glucose, we've converted it to chemical potential energy in NADH, there, and a bunch here. Then we've taken the chemical potential energy in NADH and we've used it to create potential energy in the form of a proton gradient. You'd never build it like this, would you? If you were making a system to build ATP, you wouldn't do it like this, would you? Is what you do is you build a cell which ran on glucose, because that's what goes into your body. That's unfortunately not how evolution works. So, the last thing we've got to do is get the chemical potential energy in this gradient into the form of ATP. And so there's an enzyme sitting in the membrane called ATP synthase, and that lets protons move through it. So the protons can come back down their gradient. So we keep building the gradient, but the gradient's flowing. It's like there's rain falling into a reservoir, and the reservoir gates are open. So the rain's falling into the reservoir, keeps the reservoir topped up, but as the water flows down the dam, we can use it to do some work. We can turn turbines. So the rain here is these protons coming through the membrane. So the protons come back down their gradient into the matrix. As long as we've got NADH, we can keep pumping them back. So it seems a bit silly, because they come down, you pump them back. They come down, you pump them back. As the protons come down, again, there's a release of energy. And that energy is used to make ATP from ADP. And this gives us our extra 30. So the problem we now have is that we've got electrons at complex 4. If NADH is feeding electrons in, we get a pair of electrons at 3, a pair at 4, a pair at 3, a pair at 1, and then there's like a traffic jam in the electron transport chain. You can't put any more electrons in. You've probably heard my analogy of like it's like a highway. If there's an accident at the off-ramp, you can't come on. So it all gets backed up. So you jam up the electron transport chain. So that means you've got to be able to pull the electrons out. So what we do is we take the electrons out and we give them to oxygen. If you give electrons and protons to oxygen, what do you get? You get water. Because if we add hydrogen to that, hydrogen is just a proton plus an electron. So if they combine to form a hydrogen and then you add those to oxygen, you get water. So here's my water as a waste product, and my oxygen is being reduced using the electrons from the electron transport chain. My language of reduction and oxidation is very important, because when I write questions, I will talk about the reduction of oxygen, the oxidation of NADH. So you've got to make sure you understand what it means when I say oxygen is being reduced here, and NADH is a reducing agent. That goes back to a slide I had with 4 balls on it and electrons moving all over the place. So, I've accounted for all of the energy and I've accounted for all of the matter. When we're done with this, we get another bunch of ATP, kind of around this much. Which molecule have I not yet discussed? The FADH2. I haven't talked about that, so let's finish up with FADH2. FADH2 gives us electrons into complex 2. I'm just going to write it with an F because I haven't got enough space. So the FADH2, I'll put FH2, and then here I'll put FH, FADH. So the FADH2 gives us electrons to complex 2, not to complex 1. Once they're in complex 2, they do the same thing as the electrons from complex 1. They just go through the chain, protons get pumped, and ATP gets made. Why do the electrons go into complex 2? That is because complex 1 won't accept them because it doesn't have the right electronegativity. So NADH is a little bit less electronegative than complex 1, which makes complex 1 more electronegative than NADH. So the electrons just go into complex 1 from NADH. Yeah? The end of the chain, we've got oxygen, which is, as far as we're concerned biologically, the most important, most electronegative atom. So as we move through the chain, the electron acceptors get more and more electronegative until we get to oxygen. So FADH2 can only give its electrons into complex 2, not into complex 1, because of the relative differences in the electronegativities. So that means it's got to go into FH2. You can also put other stuff in here as well. You can actually take one of the intermediates out of the citric acid cycle, and you can give it to complex 2. Complex 2 will oxidize it. So if it's got just the right electronegativity and it can be recognized by complex 2, it can grab its electron. Okay, we've accounted for the energy, we've accounted for the matter. What questions do you guys have? Yeah?
When in the electron transport chain here, where NADH turns into NAD plus in 1, is that happening in all of the other complexes as well?
So
or just that one?
NADH can only be oxidized by complex 1.
Okay.
That's because complex 1 can recognize NADH. So you know how enzymes have these active sites? It's like an enzyme. It is an enzyme. And so it will recognize its substrate, which is NADH. And so only NADH can fit in that active site. Okay, these don't have active sites for NADH, so they don't recognize it. Okay. But if NADH bumps into complex 3, it's not going to give its electrons because 3 doesn't recognize it. So only 1 can recognize NADH, and then the electrons move spontaneously from 1 to the shuttle to 3, 3 to the shuttle to 4 because of the differences in relative electronegativities.
Okay. And then the ones from FADH2 go from 2 to 3 to 4.
Yeah, they can't go backwards.
Okay.
Could they go backwards? Is there any way we could make them go backwards?
No, because it's electronegative. No,
So when it's going this direction, it's exergonic, yeah? Yeah? Energy's coming out, and the energy that's coming out is being used to drive the pumping of these protons. So if it's exergonic in this direction, that makes it endogonic in that direction, right? Is there any reason why I can't reverse an exergonic process? You can. There's a lot of bacteria which do that. There are bacteria which run this backwards to make molecules like NADH. So bacteria will actually use some electron donors to make the proton gradient, and then they've got a way of using the proton gradient to drive other electrons back up the chain to make a different type of molecule, which they need for some physiological process related to reversing the whole process. So you can. Again, welcome to Microbiology 101. Any others? Yeah?
So, FDH and NADH, they do oxidize throughout like the last step.
They will be oxidized. So FADH2 and NADH are oxidized by the electron transport chain.
And then the oxygen reduces.
The oxygen is reduced with the electrons from the electron transport chain. So you're correct, you've got slightly incomplete statements.
Okay.
So they're correct until the point where you stop, and then you've got to add in a little bit more detail. So the oxygen is reduced using electrons from the electron transport chain. NADH and FADH2 are oxidized by the electron transport chain.
Okay.
Yeah. It's complicated. It's hard to think the whole thing through. Some of these things are easy to write out because you can break them down into small pieces. It's hard to think through a complex sentence like that in your head when you've got oxidation and reduction going on. I find it easier to write that stuff out. I can only do it in my head because I've done this 4 times, 6 times this week, and I'll do it again in lab today. So if you do it that many times how many semesters I've done this for? 20. Go and do it 120 times, you're probably okay.
Thank you.
Anyone? Yeah.
I guess I'm still a little confused about the ADP and the ATP and the last part.
Can you explain what's confusing so I can...
I don't know, I just, yeah, just like how does it, like, how does the ADP get involved here?
So where does the ADP come from?
Yeah.
So your cells, they are doing a whole bunch of reactions. So what's the point of making the ATP? Why do you need ATP?
Energy.
Because it's an energy source. So your cells need the supply of energy. So when your cells need this energy, they've got ATP, yeah? We've made all this ATP, and then your cells use it, right? And what do they make when they use it?
ADP. Oh, okay.
Yeah.
So that's what, okay.
So this is all of the positive delta G stuff that your cells are doing. Doing this is also positive delta G. And we drive that using cellular respiration. Yeah. That's called the ATP cycle. We just don't talk about it. It's kind of, once you see it, you're kind of like, oh, yeah, obviously, that's where the ADP comes from. So the ADP, and then you've got a bunch of phosphate as well. So all this machine is doing is taking the energy from the proton gradient, it takes the ADP and the phosphate, and it slams them together. You know how I talk about enzymes? They take 2 things, they bring them together, and they can react them, or they can bring 2 things together and break them apart. This is bringing ADP and phosphate together, and it reacts them. Now that is, you're building, you're making, you're creating ATP there. So that's an endergonic reaction, so you've got to put energy in. The energy comes from the proton gradient. Yeah. That's weird, it's just a transformation of energies again. It's just, this is chemical potential energy, this is chemical potential energy. We're just transforming this form into this form using this machine. It's a good question though, because people don't think about where the ADP comes from. This comes from all the stuff you're doing.
Isn't that ATP, sorry to question, but is ATP going to ADP plus phosphate?
Yeah.
Is that a negative delta G or positive delta G?
Well, it's a complicated question.
Okay.
Yeah.
Okay. Never mind, I won't think about it too much.
Yeah. I mean, technically, I think is what you're saying. What you're saying is, to answer your question, yes. In my mind, it's positive, but that makes it easier for you to see, okay?
Right.
But I'm thinking about actually there's several steps, and the step I've drawn is positive.
Right.
The other piece of the step I've not drawn is negative.
That makes sense.
So yeah, you're quite. That's an easy way to think about it for you guys. Yeah. And then you take that energy and you do work with it.
Yeah.
So that's me thinking about it slightly different than you guys. We have this thing in education called expert blindness. I see things and I'm like, oh, this is so obvious. And then I write it a certain way and I don't explain to you how I'm thinking about it. It's called expert blindness and it's very difficult to overcome as an expert because it's kind of like, what do you mean you don't get it? It's obvious. And it happens all the time. So that's a classic example of that.
So then ATP to ADP, I mean ADP to ATP will be exergon then? Is that?
If you're going to build ATP, so this whole process here, yeah, making ATP from ADP, you're building, yeah? So that's endergonic. Got to put energy in. Where's the energy coming from?
The ADP? The ADP?
No, that's for making this from that. This doesn't contain enough energy to make that. You can't use the energy in this to make that.
Oh,
So
The hydrogen gradient.
proton gradient is being used to convert this to this, yeah?
oh, okay.
That's helpful. This is being built using these 2 pieces. I've got these 2 pieces. To put them together involves work. Otherwise, they're just going to sit here, yeah? So they're going to sit here. And unless I do some work, they're not going to come together. So if I put them together, that's work. Requires an input of energy. The energy is coming from the proton gradient. Yeah. If they had enough energy to bring them, to drive an enzyme to bring them together, then we wouldn't need to do all of this. You just created like the holy grail, which is a system of energy conversions which is 100% efficient, which you don't have, which is why the universe moves towards disorder and chaos. Yeah.
Does the outer membrane serve any purpose other than just keeping the protons in?
Keeps protons in. Yeah.
Okay.
Creates a compartment. Remember we talked about, well, you guys read about like that short video lecture? Compartments are important because it lets you keep things apart. Here it lets you have a relatively small volume where you can create a high concentration of something. If you didn't have the outer membrane, the protons would just float away. Bacteria actually have that problem because they pump the protons across their cell membrane. So they actually have a bit of a problem. They're not actually too efficient. They have other mechanisms for keeping them close. I mean, you think about it, the inside of a cell is negatively charged, so if you pump protons out, they actually get attracted back to the surface, so they have ways to overcome it, but they're a little less efficient than you are. We're good? Okay. You can stop now, Chloe.